Okay, so that I could use this presentation in other contexts, I started off with a couple of slides about experimental evolution. I don't think we really need to belabor the point here, how experimental evolution works. I think most people are familiar with it. You create some sort of selection pressure, you let time pass, and then you check how phenotypes have changed. And nowadays, it's easy to sequence to try to figure out what genes might underlie these changes. So that's the sort of method that we have been mainly using in my lab. And over the past, I don't know, 10 years approximately, I've been particularly interested in sex chromosome evolution. I can just go through the sort of canonical model of sex chromosome evolution briefly. So we think that you start off with a pair of autosomes, and this could be either in a hermaphroditic species that transitions to having separate sexes, or it could be in a species that has, say, temperature-dependent sex determination and switches to genetic sex determination. So the starting point could be different, but that we think that it's a similar sort of process all the way through, that you have a sex-determining locus that arises on the proto-Y in this example. This leads to cessation of recombination and accumulation of male benefit alleles around this locus. The two go together, that if you are evolving male benefit things on your Y, you probably want to make sure that they're being passed on to sons and not to daughters. So this is what causes the recombination cessation. There's been a lot of modeling of sexually antagonistic effects as driving sex chromosome evolution, but the empirical data, to be honest with you, is a little bit less clear cut whether that's always the driving force or not. This, after enough time, can lead to degeneration on the Y chromosome, because through recombination cessation, you end up, basically accumulating a lot of deleterious mutations, and you can get degradation. And this can lead to partial or complete dosage compensation on the X, which is something I'll talk a bit about later. And then over time, you could even have the Y being lost. There are some other potential dynamics here. If you have the fusion of an autosome with a sex chromosome, you get the formation of a neosex chromosome. And you could get another sort of turnover where you have a new sex-determining locus on an autosome, say, that arises. So I got interested in studying sex chromosomes because there's a lot of interesting dynamics. And I think you can kind of think of them as being potentially a model for a lot of different types of evolutionary processes. So you have this degeneration effect. You can have faster X effects, that it seems like mutations fix faster on the X chromosome. And particularly interesting from my perspective is that you can have sex-limited selection and rates of evolution, and these things sometimes go hand in hand. So I've done a number of different experiments over the years trying to understand this. We did some flatworm stuff, trying to look at the early stages of sex chromosome evolution. And then we've done some things which are more along these lines. What happens when something that was once expressed in both sexes becomes sex-limited? And so what we've done over the past few years now is a male-limited X chromosome evolution experiment. This is actually a repeat of something that I did as a postdoc. And right now, I feel like the moral of the story is never repeat an experiment, because you don't get the same results twice. But anyway, the motivation for doing this experiment. So X chromosomes being passed through the male line over and over again. So why were we interested in the X chromosome specifically? Well, the first time around I did this sort of experiment, then it was building on work by Bill Rice and Adam Chippendale, where they had taken sort of half of the genome, so all of the major autosomes and the X, and subjected them to male-limited evolution. And so we were interested initially in sort of focusing on the X because of all of these dynamics that I was talking about, as well as the fact that there are some predictions that it should be enriched for sexually antagonistic variation, so things that are good in males and bad in females, or vice versa. There's models that suggest that this should be the case. The empirical data is a little bit more equivocal, so this isn't necessarily a super strong prediction anymore. It was a stronger prediction the first time around I did the experiment, like 15 years ago. There's also predictions about the dominance of sexually antagonistic loci. So even if we don't necessarily think that the X is enriched, there could still be sexually antagonistic alleles there. And there's these predictions about dominance, that something that's good for males and bad for females should be recessive in females. because the males are going to express it anyway. And on the female side, you don't get the cost until it becomes higher in frequency if it's recessive. It's unclear as well whether we expect the X to be sort of closer to the male or the female optimum. It spends more time in females, so twice the amount of time on average in female bodies compared to male bodies. So that might make us expect some sort of feminization of the X or demasculinization, so movement of male benefit genes away from the X. But we also know that sexual selection is often stronger in males, and this could lead to more efficient selection of X-linked loci in males because of hemizygosity. So this could sort of pull the X in a masculinizing direction. There's some comparative results from flies where it seems like this might be sort of related to the age of a gene. Young genes that are good for males seem to be more preferentially located on the X, whereas old ones seem to be more preferentially located on the autosomes. And previous results, so when I did this experiment the first time around, suggested dosage compensation might be a constraint in males on the X. And that was really the motivation for doing the experiment again, is because I had gotten some results that I felt were a little bit tantalizing, that there might be something interesting going on with dosage compensation. But by that time, the fly lines were gone. I didn't really have time to pursue it anyway because I was doing other things. But I decided to come back to it and wanted to dig into that aspect of things particularly. I'm not going to have any dosage compensation results today because we've sequenced all of our samples and gotten the data back, but we've only just started analyzing it. But I will talk about it a little bit just because it was the main motivation for the experiment in the first place, or sort of rerunning it in the first place. So as I briefly mentioned before, dosage compensation, it's a mechanism for avoiding imbalance of expression between the sexes. And this is not in every single species that has heteromorphic sex chromosomes. It varies quite a lot, but some of them have it. And there's a continuum from no dosage compensation to partial dosage compensation to complete or global dosage compensation. And it seems to be somewhat random or unpredictable what mechanism different species use, because it's not sort of always the same way that it's achieved. So in mammals, for example, then in females, 1X is inactivated so that the dosage between the sexes for the X chromosome is the same. In C. elegans nematodes, then in the hermaphroditic double X individuals, both X's are downregulated a little bit so that they end up being at the same sort of dosage as the single X in males. And in Drosophila, they have kind of the most intuitively sensible solution here, that the males hyper activate their X. So they upregulate the X so that it's expressed in equal amounts as the 2X of females. So this upregulation seems to spread outwards from so-called high affinity sites, so that this green MSL complex binds to these so-called high affinity sites on the X and then causes upregulation as you move outwards from those sites. So even though we typically call it global dosage compensation, it probably varies a little bit along the X chromosome depending on your proximity to these sites. And male biased genes tend to be located further away from the high affinity sites, which presumably allows more independent control of expression. So this is kind of a complicated figure, but the main panels here are that things that are binding the male-specific lethal complex, they tend to be closer to high affinity sites. The expression bias of the genes that are at different distances from the high affinity sites, you can see that the male bias genes are sort of farther on average from high affinity sites. And when you look at this panel C, percent genes bound by the MSL complex is fewer in the male biased genes compared to the female biased and unbiased genes. So this suggests that it's bad for a male biased gene to be located close to a high affinity site for whatever reason. So this MLX experiment, as we call it, male limited X chromosome, it works like this. And this is the same protocol that we used the first time around. We're doing it again. So we have females that have a double X. So we have a more or less wild-type male that is mated to a female that has 2 X's and a Y. And as I assume you know, it's the ratio of X's to autosomes that determines sex in Drosophila and not the presence of the Y. So those females still are female. The Y does seem to be expressed a little bit in females that carry it, so it's a little bit unclear how much this messes them up. But it's a confound that I don't think we really can get around very well. Yeah, there are potentially other ways that one could enforce father-son inheritance of the X, but I don't think they would necessarily be better. Under this crossing scheme, what happens is that the X goes from father to son, the Y goes from mother to son, the double X goes from mother to daughter, and then the Y goes from father to daughter. And this will result in the production of double Y and triple X individuals, but they're not viable. The triple X individuals seem to be able to survive as larvae for a while, apparently, but they don't normally make it beyond pupation. So this is something that can be relevant, I think, when thinking about the results that we've obtained, is that even though we try to control the density in the vials, it's maybe a little bit harder to control than what it would be in the normal case, because you have to have the females laying twice as many eggs as usual, because half of them are not going to turn into anything, but maybe some of them hatch and are available or sort of competing in the early stages as larvae. So the upshot of this, or the prediction, is that the X should become subject to similar selection pressures as the Y chromosome. So basically, things that are good for males. So what did we find when we did this experiment a number of years ago when I was a postdoc? Well, one thing that we found was that one generation of father to son inheritance of the X seems to be bad. And what this treatment is showing is relative fitness as sort of competitive paternity share in the control lines, the evolved MLX lines that have been evolving for 30 generations or 40 generations or something. And then this so-called CDX are males where DX females were crossed to control males just once, and then this is their son's fitness. So these ones have lowest fitness, and they have had one generation of parental or paternal transmission of the X, and these ones, the MLX, have had multiple generations. So it seems like there might be something bad about inheriting your X from your father, at least in the short term. But this seems to be recovered over time if you continue to adapt to that crossing scheme. We don't know for sure if this is really something to do about the X being paternally inherited or maybe the Y being maternally inherited. We have some microarray data in this paper that suggests that maybe it's more about the DX moms just being kind of crappy and not necessarily about imprinting effects on the chromosomes, but we don't know 100%. The main results of interest, I would say, were that we found that after 30 generations, male fitness increased, but there was no decrease in females. So under this sexually antagonistic scenario, you would expect that an increase in males should lead to a decrease in females in their fitness because they're becoming masculinized somehow and it's bad for them. But we didn't see any difference in the females. This could be interpreted as a result of the sort of dominance patterns that I was talking about, that the things that are increasing in frequency in the MLX lines in this case are things that are recessive in females anyway, so they weren't really being expressed. We also found that locomotory activity increased in females, but not in males, so this is panel B. So the males, there's no difference, but in the females, the MLX females walked around basically as much as the males did. This wasn't something that we necessarily expected a priori. It was because the microarray data suggested that there was a lot of genes associated with metabolism and muscle development and stuff that had changed in expression. So then we thought, well, what are they doing with their muscles? Maybe they're walking around more. This is presumably driven by some sort of selection through the male side, but the fact that the males didn't show greater locomotion, maybe they were putting that energy into something else. So maybe specifically courting is something that they were spending their energy on. Expression generally changed in the same direction as sexual dimorphism. So that's what's shown in panel C here. So we took a measurement. These are only the things that were differentially expressed as a result of the selection treatment. But if you then classify them according to their fold change between the sexes, so that you have male biased stuff on the positive side and female biased stuff on the negative side, then you can see that overwhelmingly it's going in the same direction. So stuff that is male biased was becoming upregulated in MLX, and stuff that was female biased is becoming downregulated in MLX. And so this is like, yeah, of 480 genes or something, 450 of them were going in the same direction. And it seemed like there was some association with dosage compensation, which was, like I said, the motivation for deciding to do this experiment again. So it wasn't something that I had expected a priori, but I had started getting more interested in sex chromosome evolution in general and been reading about dosage compensation. And I thought to check, okay, does this distance from the high affinity sites seem to be associated with anything? And for the upregulated genes, then there was a positive association. So things that were further away from the high affinity site were upregulated more. We don't really know what's cause and effect here. It could be that they were being more upregulated just because those were more likely to be male biased genes, the ones that were further away. But that being said, I mean, the fact that they changed after changing the selection pressures on the X suggests to me that if they had sort of wanted to be more upregulated, they could have done so if it wasn't for counter selection in females. So in order to dig into this into a bit more detail, we decided to do this experiment again and basically, really double down on the genomics part a bit more so that we could really try to look into the dosage compensation aspect more. So in the current run of this experiment, we had 3 replicate populations within each selection treatment, the same as last time. The previous 2 big experiments that I'd done before this, in the flatworms and in the flies doing a female limited X chromosome experiment, we had 4 replicate populations. And after doing that a couple of times, I decided that 3 was enough because it didn't feel like it really increased our power very much to have 4, and it just made everything more work. So we decided to stick with 3. I don't know if that's a good or a bad idea, but anyway, it felt like it was the minimum to be able to say that something is consistent. We measured fitness every 20 generations because we basically just wanted to establish that we saw a similar response as last time. We didn't really plan to do a lot of phenotypic assays, but then we, decided to focus, like I say, more on the genomic data. So collecting RNA-seq and whole genome sequencing data, pool-seq, every 10 generations. We didn't, yeah, okay, I'll save that for in a minute. So we also collected different female samples this time around. So last time around, what I called the sort of female MLX in those figures were actually individuals that had half of their genome coming from the MLX line and half of their genome coming from the control line. So females that were basically heterozygous between the 2 lines. And we did this because we were worried that there might be sort of inbreeding effects or something on the X that could make those females perform badly, even if there weren't actually sexually antagonistic effects. So we decided to go the safe route and only look at the sort of heterozygous females. This time around, we decided, once we decided to collect more phenotypic data, that we would have both types of female genotype. So both these sort of heterozygous females that are crossed between the MLX flies and the control flies, and pure MLX females that have all of their genome from the MLX lines. And this way we could look at some of these predictions about dominance. We didn't really find what we expected when it came to fitness. I might as well spoil that right now. And so in the end, we decided to measure a bunch of other traits just so that we could, try to figure out what was going on and see if we saw changes in other traits. So at the end of the experiment, because we're not maintaining the lines anymore, we measured egg to adult survival, locomotion, mating latency, offspring sex ratio, thorax size, and male fertility. A couple of these we had measured more than once, locomotion and mating in particular, but the results were qualitatively similar at the early generations compared to the later ones. So I'm only going to present the later one. And we terminated the experiment after about 70 generations. One other thing that I could mention that I don't have a specific slide for is that I'm sure some of you have noticed that if you just do this over and over again, you could have clonal evolution of the X's because they're never getting the chance to recombine. So we included the so-called recombination box that Bill Rice had when he originally developed these type of experiments, where 10% of the population in the MLX flies are moved into this sort of recombination box where X's can go into females and recombine in female bodies and then be moved back into the population. And that seems to be sufficient to avoid a lot of hitchhiking effects, at least. Not necessarily everything. Okay, so fitness results. When we do fitness, then we typically measure female fecundity in an 18-hour window, which matches the window of laying time that we use in our standard sort of maintenance protocol. And for males, we usually do a red eye, brown eye competitive fitness assay, where the brown-eyed competitors compete with the red-eyed target males for matings with brown-eyed females, and then all of the red-eyed offspring in the next generation, came from the red-eyed target males. So we know that the females like the red-eyed males a little bit better than the brown-eyed males, but because we're always comparing basically red-eyed progeny from MLX with red-eyed progeny from control, that doesn't really matter. So we found lower male fitness at generation 20, potentially consistent with a deleterious effect of this DX or the DX moms or some sort of adaptation to the presence of the DX. I'm not going to say that this was a really strong prediction that we expected to see a decrease after so many generations. It was more that after the fact, we realized that it could be a possibility because I had done more generations of back crossing the first time around of the DX strain into the, sort of standard base population, so that maybe there had been some adaptation going on already before we sort of officially started the experiment. So at generation 40, we saw no difference in male fitness between control and MLX. And these are standardized fitness measurements, both so that we can compare them between generations and also between the sexes. So the control is always set to 0, basically, and then the MLX is scaled compared to the control. And so we thought, okay, great. So they did worse at first, but now they're recovering. And if we do this for another 20 generations, then they're probably going to have higher fitness. Can you guess what's coming? No, it decreased again at generation 60. We were like, what? Why? Oh, so annoying. So we have this weird kind of hump-shaped pattern then where they're doing worse and then fine and then worse again. And if we accept that some sort of confounding effect of the DX that they hadn't fully adapted to at generation 20 is the explanation for this, we're still left wondering, well, why do they do bad again at generation 60? Female fitness was never significantly different. The first time we measured female fitness, we only measured 2 of the genotypes, this control and then the sort of heterozygote, and then we didn't measure the MLX one. But there was never any difference in the female genotypes. So this is kind of puzzling because we did have some sort of a priori expectation of how things should potentially change, right? And I would say that taking the male and the female fitness results together, we don't really have any clear-cut support for any of these explanations. So if we were thinking sexual antagonism, then we were thinking, okay, things that are good for males and bad for females are increasing in frequency. So if male fitness goes up, female fitness should go down. We didn't see that. But even if we sort of accept the sexual antagonism explanation, but assume that the males are doing worse for whatever reason, then you would expect the females to do better. But there wasn't any change. So that doesn't really fit. If the fact that the X is going through males is having some sort of beneficial effect on purging of deleterious alleles for whatever reason, then that could result in increased male fitness, but then you would expect to see a correlated response in females. And if it was just an effect of drift, that the males are doing bad because of drift, then you would expect the females to do worse as well, which they're clearly not. So even though we know, of course, that drift is going to be happening, that doesn't really seem like a super convincing explanation for this change in fitness that we see at generation 60. So because of this, we decided that we would try to measure other traits and see if we could get any insight into what might be going on. And basically, is anything happening? Are they getting worse? But what, are they changing in any other way that might seem to be adaptive? So egg to adult survival, we measured, and there was no difference. This is only for male parents, because then we know that the X from the MLX father is going to end up in both sons and daughters. And there was no difference there. Survival was really high and basically identical between the two treatments. When we looked at locomotory activity, then we found that male locomotion was higher than female locomotion, as we might expect, but that there wasn't any significant difference between the selection regimes. So if anything, MLX is a little bit less active, but it's not significant. Somebody asked me yesterday about the difference between the sexes here, that it was kind of small. And I would have to double check how we ran this experiment, but it might be because previously we had them in groups and this time we had them singly. I think that might potentially be the reason. I would have to double check to be sure. We also looked at mating latency, and this was sort of strange because there was no effective selection regime, but there was a difference between the sexes. So these are these MLX females that have the sort of double dose of the MLX genome, and they're always mating with a member of the ancestral population in these experiments. So these are evolved females with ancestral males and evolved males with ancestral females. And the male mating pairs, or the pairs where the male was the target fly, the mating latency is higher. So this to me is suggesting that either the females in both the control and the MLX lines are somehow becoming less choosy than in the ancestor, or that the males in both the selection treatments are somehow worse than the ancestor. This could be some sort of effect of smaller effective population size. We've done some experiments following up on this to see what the sort of baseline was in the ancestral population, how fast they mate, so that we can tell whether it's that the females have gotten faster or the males have gotten slower. But that isn't completely analyzed yet, so I haven't been able to do the comparison. But that was a little bit strange anyway, and potentially worrying if the fact that we reduce the effective population size a little bit is having such a big effect. Offspring sex ratio is something that we looked at because I looked at this in the sort of original run of the experiment, and there was really no difference at all. You can see that they're almost exactly the same. And so this is the parental sex. So if the mom was the target genotype or if the dad was the target genotype, and it doesn't make any difference. And then finally, we found something kind of interesting when we looked at body size, and very unexpected, I would say. So the degree of sexual dimorphism in body size seems to have increased in the MLX lines compared to the control lines. So on the left, you have females, and so this one is control, this one is MLX. On the right, you have males, control, and MLX. And so the females are going up a little bit in body size, and the males are going down a little bit in body size. This is odd in several ways, I would say. Both the fact that they're going in opposite directions, which is not something that we would expect under a sort of correlated genetic effect scenario. The males getting a bit smaller is something that we've seen in the previous ML experiments that were done by Adam Chippendale's lab. There does seem to be some sort of opposite selection for body size, at least in the base population. So the fact that they're responding like this is kind of an adaptive response, but we don't know why it's happening. And we don't really understand why the females would be getting larger. And then finally, because we didn't see the sort of expected pattern with fitness, but something seemed to be changing, then we started thinking a little bit about, okay, well, maybe is this something to do with the fact that the DX females need to produce twice as many eggs to get the same number of offspring, so maybe sperm limitation could actually be a thing. And so maybe the MLX males are investing more in, say, sperm number and trading it off with some other aspect of sperm competitiveness. So maybe investing less in SFPs or something. Or potentially that the DX females are just a bit crappier overall, so it's not good to be too harmful to them, so that they invest less in competitive traits. So to try to test this hypothesis, we wanted to do a bit of a quick and dirty assay. We didn't really think there was much point in doing a sort of P1, P2 with paternity as the first male to mate versus paternity as the second male to mate, because we already knew that they were doing worse than the competitor or than the control. So instead we thought, well, okay, if we have a no contest situation where we just mate a single male and a single female once and then let her lay eggs until she runs out of sperm, do we see a difference? And it's not super convincing because it's only P0.06, but it does seem to be the case that the MLX, which is the red here, can maintain offspring production a little bit longer. So females mated to MLX males. And this translates to, if you just look at the sort of number of days that they lay fertile eggs and the total number, it's about 30 more eggs in total, and it's about 1 more day of laying. So it's not like it's a massive effect, but that would at least be consistent with our hypothesis. Okay, so the female genotypes that we measured, we were interested in looking at dominance effects. And regardless of whether we think that there's a large effect of sexual antagonism or not, we still thought this would be a good opportunity to just look and see, do we see an overall pattern of, say, dominance in specific traits or even non-additivity. Presumably this would be sort of the net effect of many different loci, or if you had a few large effect loci and they were all sort of going in the same direction, then you would be able to pick up a pattern. So we collected data for female fitness, locomotion, and thorax size. And the plots that I'm going to show after this are basically set up like this, so that you have the control mean, the MLX mean, and then this heterozygote treatment. And the dashed line is the sort of additive prediction. So taking the mean between the control and the MLX, you would expect the MC treatment to end up on with the mean on that dashed line or dotted line. And in this case, locomotor activity, it was not significantly different. All the previous figures that I've shown, the error bars have been standard errors, but these ones are confidence intervals, 95% confidence intervals, so that if they overlap, you can say that it's non-significant. We tested, oh yeah, and I didn't mention that before, but we tested both overall and in each replicate population. So we've done a similar type of approach in a previous experiment, and we could see for some traits that it maybe looked additive overall, but then when you looked at each replicate population separately, you could see that, well, 2 of them, it looked as if it was recessive, and in 2 of them, it looked as if it was dominant, and then when you average those, it looks additive. So we did both. We looked both sort of overall and within each replicate population. And the replicate populations are otherwise, the numbers don't really mean very much, because number 1 in MLX is not the same as number 1 in control, except for in this context, because replicate 1, the MC treatment is a cross between the other 2. So it seems like there might be some evidence of dominance effects in thorax size, and this is consistent with data that we've collected in another context. So that's kind of reassuring. The overall p-value is only 0.06, and this is sort of the overall pattern where the MC treatment seems to be more similar to the MLX treatment. And when you break it up into the replicate populations, it's significant in rep 1 and rep 3, but the pattern is a little bit different. So in rep 1 here, this is almost looking more like overdominance. In rep 2, it's looking more like the control variant is dominant, or it's looking sort of recessive in MC compared to MLX. And in rep 3 is sort of the most convincing signature of dominance rather than anything else. But overall, it seems like there's non-additive effects happening in body size. Okay, So we have a grab bag of results here. They're not the same as what I got before. And I have to say this is extremely frustrating because this was one of the experiments in my career where I was like the most convinced that we would be able to repeat it successfully, because we accidentally reran the experiment in between when I did it as a postdoc and when I did it now. So a number of years ago, we did an experiment where we took X and Y chromosomes from different population origins and put them together. And that's probably the other experiment where I was most convinced I would get a particular result, and I didn't. But what we saw there was that if you had a mismatched X chromosome compared to the rest of your genome, or a mismatched Y compared to the rest of the genome, then you actually had higher male fitness than if you had matching. And we assume that this must have some sort of cost to it, because otherwise those variants would spread everywhere. So we did an experimental evolution sort of follow-up to that, where we let these genotypes that had a new X or a new Y just evolve for 20 or 25 generations and then tested their fitness again. And the first time around that we did it, we used the DX females for one of the genotypes. And we saw that in the genotype that included the DX females, the higher fitness compared to the wild type was maintained. But in the one where we didn't use the DX females, it was lost. And so at the time, we thought that was a big goof because it was a bit silly to not have thought of that. But it convinced me that this actually was something that was repeatable, because we had seen, oh, you use the DX females and you're enforcing father to son inheritance of the X and you have high fitness. So I was pretty convinced that this was going to happen again, but it didn't. So where are we now? Well, some potential explanations here. When it comes to the male fitness results, then I think this is probably something to do with altered sexual conflict dynamics. As I mentioned before, either the fact that the DX females are maybe just a little bit more fragile, or less fit than the wild type females, so that this then selects for males to be less harmful, and then they become less competitive. It could also be some sort of trade-off between sperm number and sperm quality, or other aspects of competitiveness, to do with the fact that so many eggs are being produced that aren't going to end up developing. The increase in sexual dimorphism result is very strange. And the only thing that I can think of that might explain that is this modeling paper from last year by Muralidhar and Koop, where they were looking at the polygenic, the response to selection on X's and autosomes under sexually antagonistic selection with polygenic architecture. And this is from the no dosage compensation scenario, but in that model, that basically just corresponds to sort of asymmetric selection pressures between males and females on the X. And what they found was that if you have some sort of phenotype and you're selecting for smaller phenotype values in females and larger phenotype values in males, so selection going in opposite directions, if you track the net effect in their simulations, then you get from your starting point to sort of the optimum for both sexes. But when you disentangle the response into the X component, which is the orange thing here, and the autosomal component, which is the green, then it becomes clear that the X, because it's spending more time in female bodies basically, and it's being exposed to more female selection, is sort of dragging the phenotype closer to the female optimum. And then the autosomes are pulling back a little bit. And that's what's shown on the right here, that autosomal loci tend to be sort of biased towards the male additive genetic value, and X-linked loci tends to be biased towards the female genetic value. Because we reversed the selection pressures on the X in our experiment, then maybe this is what we're seeing, that the X was sort of contributing loci for smaller male size, and that the autosomes were compensating so that the females weren't changing in size in the experimental populations. But then when you put these into, put them into a new female body that's not a DX female body, then maybe you can sort of see the effect of the autosomes overwhelming the effect of the X. I don't know. It's a speculation. I think it would be interesting to follow up on this. I'm not sure how much we'll be able to do it with this particular experiment, but we do have data from a previous experimental evolutionary experiment where we actually try to disentangle the effect of the autosomes and the evolved X chromosome. Okay, so conclusions. Results are not really consistent with previous data. We basically don't know why, unfortunately. Predicting responses to selection is hard, which I'm sure is no news to most of you. But even still, a little bit surprising. I mean, one potential explanation could be that the starting population was a little bit different. So the first time around I did the experiment, I used Adam Chippendale's LHM-based population as the starting point. And this time around, it's LHMs that came from Ted Morrow's lab. And there is some sequencing evidence that suggests that they are a little bit different. That being said, I don't know how much I believe that explanation, because the sort of second time we accidentally ran the MLX experiment, that was using lines from Ted's lab. So if it worked that time, why didn't it work this time? Who knows? I also don't really think that there's been a lot of drift or a lot of changes happening within our group, over the years, because we just recently did some extra data collection to have a sort of baseline of our ancestral population, and comparing the same traits that we measured 10 years ago versus now. And for example, female fecundity is exactly the same. And things like locomotor activity and mating latency are almost exactly the same, despite those being pretty noisy traits. So it doesn't seem like there's an awful lot happening, at least on the phenotype level. We're hoping that the genomic data that we collected will provide better clarity, because some of these explanations, even though, say, this kind of trade-off between sperm quantity and other components of male fitness, is hard to really say anything from the phenotypic data we have at hand. We should be able to say something when we look at expression data. And we're still interested in this dosage compensation aspect. All of the fitness stuff was sort of intended to be just a minor aspect of this project. We basically just wanted to confirm that, yes, we had a similar response as last time, and then we could really focus on the genomic data, and we plan to do a TAC-seq and a bunch of things. And then because we didn't get the expected phenotypic response, that left us sort of scratching our heads and feeling like we needed to do what we could to try to figure that out. But now at least we have some hypotheses that we can maybe try to confirm or disprove with the genomic data. And in some sense, it doesn't really matter, actually, if the males are performing better compared to our standard competitor. If we look at the dosage compensation machinery and see that there are changes going on there, or if we see a similar pattern of changes in regulation depending on distance to these high affinity sites, then we can still actually investigate that hypothesis, regardless of whether we can sort of understand the phenotypic results really easily or not. So we'll see. We've only just started analyzing the expression data. And yeah, there are things that are changing. That's pretty clear. It seems to be unevenly distributed across the generations, which is a little bit worrying. I'm not really sure why generation 40 seems to be so different than the others. But anyway, it's preliminary right now, so we can't really say. So that was basically everything I wanted to tell you. So this was, as I said, mainly work done by my PhD student, Demodeep, and then with help from Kat, who's a researcher in my lab and runs the lab while I'm head of department. Thank you.